Mhm.
Subuh nanti subuh ya. என்ன என்ன நம்ம பிரின்சிபல் சாரா? சரிப்பா, ஓகே.
మామూలు స్టూడెంట్ ఎగ్జామ్ అనమాట. ఇక్కడ ఉంటాయి రికార్డ్స్, ఓల్డ్ రికార్డ్స్.
സാരില്ല, അവര് പോകുമ്പോ എന്നെ ഇറക്കി വിട്ടാൽ മതി.
Start.
Okay.
Good morning, one and all.
Good morning, ma'am.
Good morning, one and all. As they say, the future of medicine will not be discovered by humans or machines alone, but by their intelligence working together. A very warm and cordial welcome to one and all. It is my privilege to welcome you to the 5-day online faculty development program on artificial intelligence in transforming drug discovery research, conducted from 27th July to 31st July and organized by Center for Advanced Molecular Design and Simulation. I am J.E. Rachel Nivedita, research scholar from the Department of Pharmaceutical Chemistry, and it is my pleasure to be your host for today's session. Without further delay, I now invite Dr. Jubi Selvaraj ma'am and take the session forward. Over to you, ma'am.
Thank you, Miss Rachel. Yeah, a very warm good morning to all the fellow teachers who have joined virtually. So today's speaker, Dr. Chandan S. and his team will join very shortly. Within 10-15 minutes, they will be joining. So before that, I will be giving a brief introduction about the integration of artificial intelligence in network pharmacology. So let me share my screen. Rachel, is my screen visible?
Yes, ma'am.
Yeah.
Yes, ma'am.
Yeah. Thank you, Rachel. So I'll be briefly discussing about the introduction, why this network pharmacology. And we all know the network pharmacology, the basic need of network pharmacology is big data era. And how do you connect network pharmacology into polypharmacology? And what is this network pharmacology approach? And what are all the parameters we have to look into? We have to look into nodes, edges, and hub genes and the measures, okay, the network centrality measures, applications in drug discovery, challenges and future perspectives and conclusion. Yeah. So before going to this network pharmacology, let me briefly talk about the drug discovery revolution into 3 phases. So the first phase is the classical model. That means it is the random screening or what we can say, it is a serendipity, discovery of drug, accidental discovery. Okay, and without knowing whether this molecule will give a defined activity or not, people used to synthesize or people used to isolate it from the natural source. So that is the classical model. So the very good example, no, it is Sir Alexander Fleming's accidental discovery of penicillins. Okay, like that, majorly whatever drugs we have in the market, no, all are discovered through this serendipity discovery only. And the second phase is from the traditional method, that is once the computers have come into the picture, that is after 1980s. No, before that computers have been used only for the data storage. After once the computers have been brought to the research, that's what the computer aided drug design, that field has been emerged. So it is basically based on the targets based drug discovery. Okay, one disease, one target, one drug. Okay,
And greet our distinguished guest.
yeah, has dominated the drug discovery approaches. So here again this also has got a problem. Okay, if we are focusing only on one particular target for a complex disease such as neurodegenerative disorders like Alzheimer's, or you take rheumatoid arthritis or cancer, they involve multiple targets. Okay, so if you target one particular, if you focus one particular target, again there will be some failures. Okay, resistance will occur, toxicity will occur. So slowly the target expansion, no, it has come to the broad spectrum, that is multi-target drugs have gained considerable interest in the last decade because of their potential to treat diseases involving interactions of multiple genes. Okay, so when you take a particular compound, okay, your interest, it should be mainly focus on the genes which are in multiple genes which are in the particular target. Okay, so this yielded the new era which is called network pharmacology. Okay, the one drug, one target, one disease approach. Okay, it faces many safety, efficacy and sustainability changes. So that's what the new era has come. Okay, you can see the picture. All these are the proteins or genes which are involved in the particular disease. So the connections, you can see here, there are so many connections, P7 to P8, P1 to P9. So these connections, so this particular field, it focuses how the various genes or various proteins are connected or they are modulating. Okay, they are connected in such a way they modulate the disease. Okay, so that's what the network pharmacology has emerged, which attempts to understand drug actions and interactions with multiple targets. So it uses computational power to systematically catalog the molecular interactions of a drug molecule in a living cell. Yeah. So for all these, we need the big data. Okay. You need, of course, you need the genomics, you need proteomics, omics play a major role. You need transcriptomics, you need metabolomics, everything. So it all started with big data. So over the last few decades, technological breakthroughs have permitted the collecting of massive volumes of data, resulting big data era. So the big data revolution is expected to significantly impact many parts of the health sector, including the ability to assess an individual's risk of developing sickness and improving personalized disease. Okay, as we are into this AI era, so for doing all these AI and ML tools, particularly ML tools can be very well connected to the drug design and the drug discovery process. So for all these, you need the big data. So it is the starting point of this network pharmacology. Okay, again, this network pharmacology is not a new one. Okay, it is coming from polypharmacology. So what is again polypharmacology? It is a field of study related to design, discovery, synthesis and use of pharmaceutical agents. Okay, so if the pharmaceutical agents may be drugs, small molecules, antibodies, stabilized peptides, natural products, etc. that act on multiple targets. So here again the multiple targets. So that is very, very important. So it may be a single molecule, but it should have effect on the multiple targets. So from this polypharmacology, only the network pharmacology has been evolved. Okay. So this particular network pharmacology, it suits for the neurodegenerative diseases and cancer. Okay. Where these are all the complex disease we will tell. Of course, they involve the complex biological system. So which involve the involve networks of multiple genes, hence necessitate pharmacophores as a part of their therapeutic and system biology-based approaches. Okay, so this is one particular example for your multiple pathways. So this is one particular disease which is called as rheumatoid arthritis. So this particular disease, if you take the molecular pathway, it involves so many pathways starting with Notch signaling, SYK signaling, P13K, JAK signaling, WNT signaling, MAPK signaling. So you have various genes are involved, correct? So for example, during inflammation, so many cytokines are, their level will be more. If for instance, IL-6, interleukin 6 is evolved, immediately it activates the JAK-STAT pathway. Okay, so what is the message over here is such a complex disease, it is not confined to any single pathway. It is confining to multiple pathways. Okay, so that is what I wanted to convey. So what does this network pharmacology? Okay, so what it does, it needs the data. Okay, at first it analyzes the targets from different resources, including experimental data, public data available in databases, and then performs the network analysis. Okay, so based on these networks, so here you can see the picture. It collects the, we have to collect the data. It may be from genes, it may be of metabolites. Okay, then we should do the protein interaction. Okay, you have so many types. The major type what we look into this drug discovery aspect is protein interaction, we shortly call as PPI, and protein drug interaction, PDI we call. Okay, so we have to collect the data, and we have to load into this particular platform, then we do the network analysis. So once you do the network analysis, what is your outcome? It identifies the hub genes. So what are the hub genes? The hub genes, they control the disease. Okay, they are the major genes which regulate the pathway that leads to disease condition. Okay, so you have to do the network analysis, then you have to validate, and you have to finalize the data.
We'll make sure that
So as I told you, hub genes. So hub genes are genes that have disproportionately high number of connections within a biological network. Okay, it may have so much of connections. Okay, it control the particular disease. Okay, so these networks can represent various biological processes such as gene regulatory networks, protein interaction networks, or metabolic pathways. Okay, so in simple terms, the hub genes, they act as central nodes. That's what I told, like major traffic intersections. Okay, wherever it will be controlled. Okay, likewise, this particular hub gene, it controls the various genes which are involved in the particular network. Okay, so examples, the notable examples are, very well example is our TP53, that is tumor protein 53. It is a well-known tumor suppressor gene, and it regulates the cellular stress response. Okay, it is a hub gene which regulates the cell cycle arrest, apoptosis, and DNA repair. So we know very well various cancers, they are associated either in the absence of this TP53 or mutations in the TP53. The another one is MYC. It is also a proto-oncogene. Then AKT1 serine threonine kinase. It is a very important regulator of cell survival, growth, and metabolism. Okay, and TNF, tumor necrosis factor, which is a very important factor, which regulates the systemic inflammation. So what is the message over here is they control the other genes also, or in simple way, they control the disease. Yeah. And coming to the second important things is nodes and edges. So these nodes are called vertices. These are the points or entities in the graph. Okay, you can see here all the dots, no, they are the nodes, they are the genes. Okay, and the connection, the lines are called as edges. These are the connections or relationship between nodes. Okay, so in a simple language, in a social network, each person is a node, and each person, how he connects with other person or how he makes friendship with others, so that is called as edge. So in shorter way, these are the representations for nodes and edges. This is one typical network which is involved in osteoarthritis. So this particular network, no, it has been developed from our research group. You can see here this MMP9 matrix metallo protein, okay, and prostaglandin synthase 2. So likewise, you have so many ESR1. So you can see here, these nodes, no, these are called as nodes. Okay, each one of these spheres or nodes, and these lines which are connecting these spheres, no, these are called as edges. Okay, so and again, these node, after this nodes and edges, no, we have some centralities, degree centrality, betweenness centrality, and closeness centrality, that I will discuss in my upcoming slide. This is also one more such network which has been developed from our research group. Okay, yeah, as I told you, the first one is HubG, and the second one is always we have to look into nodes and edges. And the third thing is the results. Okay, so the results, no, these three centralities plays the major role. Okay, so it will give you some mathematical values. With that value, you can find out, and you can find out the hub genes, everything. Okay, so in network analysis, understanding the importance or centrality of nodes within a network is very crucial. There are so many centralities, but we always look into these three centralities: degree, betweenness, and closeness. Okay, so what is degree? It is very, very simple. The degree centrality measures the number of direct connections a node has. So it is the simplest measure of centrality. Okay, so that means how it has connections to other. For example, for instance, if you take this MMP9, it has connection with PTGS2, it has connection with MMP2, it has connection with IGF1R, it has connection with ESR1. Likewise, how many connections the particular gene makes? So that is called as degree. Okay, so that is again a mathematical value. It is a mathematical number. Okay, yeah. So for our easy understanding, imagine a social network where nodes are people and edges represent friendships. And coming to another one is betweenness centrality. So here, this betweenness centrality gives you, it quantifies how often a node lies on a shortest path between two other nodes in the network. That means how it can reach to other two nodes, how a particular gene can act as it is like a betweenness between two nodes. Okay, so this is betweenness. And the third one is closeness. Closeness is very important. It measures the average distance from a node to all other nodes in the network. So the nodes with high closeness can quickly reach all other nodes. So for your easy understanding, I have taken 5 nodes starting with A, B, C, D, E. These are the 5 nodes, and these are all the different centralities. Okay, you can see here A, that is a particular gene A, it makes 2 degree. That means it can make only 2 connections. B can make 3 connections. C can make 2. Likewise, D can make 3 and E can make 2. And betweenness also, how often it is lying between 2 nodes. Okay, so shortly, degree means how many direct connections a node has. Betweenness means how often a node appears on the shortest path between other node, how close a node is to all other nodes. Okay, so these are the three important parameters. Yeah, so this is one particular example. Again, I have shown you the network. Here, this is the representation. This is the representation of the degree, betweenness and closeness centrality. So here, the matrix metalloproteinase 9 and almost the second B-cell lymphoma 2, no, they can be considered as hub genes because they are making the connections with more genes within the particular network. Same thing we have to look into the degree, betweenness also and closeness also. So these three parameters are very, very important when you look into the particular network. And these are the different types of networks: metabolic networks, drug interaction networks, drug disease interaction networks, protein-protein interaction networks, and isoform networks. Yeah, so this is the short explanation where and all you can integrate the artificial intelligence and machine learning in network pharmacology. So as I told you, we have clustering, we have deep learning, we have graph neural networks. So these are all the things you can integrate into the network pharmacology. Okay. So for the want of time, I'll be quickly moving through within another 5 minutes. I'll be finishing it. So you can see here all the steps. If you take all the steps in network pharmacology, each and every step, the different AI and ML tools are involved. Okay. Suppose, for example, the data collection, you need the natural language processing. Okay. For example, multi-omics, PubMed mining, like that. If you go for drug target prediction, you need all these tools, standard vector machine, deep learning, okay, random forest, all these tools. Then you convert the network. Of course, you need the clustering, the grouping, the principal component analysis, okay, where the important genes only you will be making into a group. Then you do the network analysis, and we always go for the mechanistic insights, and you have to do the validation. So always we should not stop over here. It should be validated with the further structure-based drug design methods like molecular docking and dynamics, and further it should be validated with the in vitro analysis. Okay, so these are the various steps which are involved. As I told you, how the AI and ML which are in the network. And finally, the applications. So you can very well apply in your students' projects, final year B Pharm as well as your first year M Pharm, before doing your structure-based drug design, before doing any visual screening or molecular docking, you can do this. Okay, and it is of course, it is used in rational drug discovery. It is used to identify the novel target. It is used in repurposing and data driven prioritization. It is used. Okay, so by this, I think I'll be completing some introduction part about the network pharmacology. So if you have any doubts, you can clarify with me, or you can keep it in the chat box also. Thank you all. Thank you all for your patient hearing.
Thank you, ma'am, for your valuable insight on drug discovery and network pharmacology. It is now my privilege to invite Dr. Chandan Shivamulu, sir, to deliver today's lecture on artificial intelligence-driven bioinformatics approaches for modern drug discovery, from target identification to drug repurposing. This session promises to provide valuable insights into how artificial intelligence and bioinformatics are revolutionizing the drug discovery process, from identifying novel therapeutic targets to accelerating drug repurposing. Over to you, sir.
Thank you,
Yeah,
ma'am.
Rachel. Yeah.
Yeah.
Sir, good morning, sir. Chandan, sir.
Good morning, Jubi, ma'am. Good morning.
Yeah,
My
good
voice
morning,
is
sir.
clear, ma'am.
Yeah. I will be introducing you, sir. Then you can start your session.
Thank you so much,
Yeah.
ma'am. Thank you.
Yeah.
Thank you for that.
So again, good morning, teachers. So it's my heartful pleasure to introduce Dr. Chandan S, who is a dynamic personality and Deputy Dean Research in JSS Academy of Higher Education and Research, Mysuru, Karnataka, India. So Dr. Chandan S, Deputy Dean, he has 16 years of academic research and administrative experience. He specializes in molecular biology, microbiology, bioinformatics, and virology. He holds a PhD in biotechnology with a research focus on molecular diagnostics for leptospirosis and have been associated with national projects at ICAR and ICMR. His scientific contributions include over 270 research publications, multiple national and international patents, and editorial roles with journals like Nature Scientific Reports, Frontiers, IJHAS, which is our in-house journal. He is recognized with the DST SERB Young Scientist Award, multiple Best Researcher Awards, and international fellowships. He has consistently ranked as the number one researcher at our university, that is JSS Academy of Higher Education and Research, based on Scopus and Web of Science outputs. Through Scival metrics, his research has demonstrated a field-weighted citation impact of 1.16, which is a promising factor, surpassing global averages with over 3,200 citations and an H index of 31, reflecting both depth and visibility. His work spans multidisciplinary domains such as biochemistry, medicine, pharmacology, chemistry, environmental and computational sciences, bridging research and translational impact. Driven by a vision to shape future scientists, he is committed to mentoring research scholars, developing sustainable innovations, and advancing India's global research presence in life sciences and health technologies. So, with this brief introduction, now I invite today's speaker, Dr. Chandan S, for his valuable session. So, the session over to you, Chandan sir. Yeah, the
Thank you,
screen
Jubi,
is
ma'am.
yours.
Thank you so much, ma'am. Thank you for that introduction. Ma'am, with your permission, I'll share my slides. Like maybe if you can close the slides,
Yes, sir. Yes, sure.
share my slides.
Yes, sir.
Yes, ma'am. Thank you. Ma'am, is the slides visible? Slide visible, ma'am?
Yes,
Yes, sir. Yes,
yes,
sir.
Yeah, thank you. Yeah, maybe with this, I'll start my today's session. Thank you so much. It's a good morning. First of all, good morning to everyone there, and greetings from JSSAHR Mysuru. And as I introduced, I work for the research division at JSSAHR. It's my great pleasure and privilege to be a part of this a 5-day FDP program, which is happening on AI, particularly on artificial intelligence. Maybe when it connects to the pharmaceutical research organized by JSS College of Pharmacy UT. I sincerely thank the opportunity given for me, especially to Dorai Anand Kumar sir, for providing me this opportunity to present my work or whatever the thing which I know. share my thoughts on the topic that is rapidly transforming the healthcare, which we are seeing it today. And basically, when we think about the AI and bioinformatics, which when we talk about the approaches in clinical trial design and drug repurposing, we are currently like we are witnessing an exceptional maybe we can say convergence of artificial intelligence, bioinformatics. Maybe we can also think about the bioinformatics, big data analytics, or the clinical studies also which can be considered. And these techniques, when we see these techniques, are not only the accelerating the pace of biomedical research, but are also like identify and eligible the whatever the patients are seeing the problems or predicting the outcomes and discovering the new therapeutics opportunities from the whatever the existing drugs are there. That is what I have opted for today's topic, that is drug repurposing. In today's presentation, I'll be discussing more about how AI-driven approaches and bioinformatics tools are helping researchers and make the clinical trials more efficient and cost-effective and patient-centric, while simultaneously enabling the faster drug repurposing strategies, we can say, that can shorten the journey from discovery to, we can say, the patient care. Ultimately, when we whatever we do, it is ultimately for the patient care. I hope this session will definitely provide the useful insights into the emerging role of computational intelligence in modern healthcare research and stimulate. Definitely, it stimulates the new ideas for your own research, whatever the endeavors which we are all doing. With this, let us begin our journey into the existing world of AI in bioinformatics in clinical trial design and repurposing. Now, before I start, I would like to briefly tell about our institute, because I heard from Anand sir that is many of the faculty members are from the other institutes. That's why before starting my topic, I would like to take maybe a tour, what and all the facilities we have and where we are. These are the things which I'll be briefing to the participants. Before I start my presentation, taking the blessings of His Holiness Jagadguru Dr. Shri Rajendra Shivaratri Rajendra Mahaswamiji, where he taught us the ignorance is the root of all the evils in the society and education is the like a sovereign remedy. This is what our main motto of our Swamiji. And pranams to our present chancellor, taking his blessings, His Holiness Jagadguru Shri Shivratri Deshikendra Mahaswamiji, the present pontiff of our institute, the 24th Peetaध्यksha of Jagadguru Veera Simhasana Mat, Sutturu Shri Kshetra. With this, I would like to take through the research thrust areas where JSS-AHR is working towards. When we talk about the different thrust areas where we work is maybe few of the faculty members are working towards the stem cell and regenerative biology, and also nanomedical and biomaterials, cancer biology and biomarkers, and public health and community health, disease biology and biospecimen research, and infection diseases and diagnostics. And one of the major area which we are all talking today, drug discovery and pharmaceutical sciences. And a few of the faculty members are working towards the domain environmental and health and data sciences. And these are the different thrust areas which most of the faculty members are working towards. And when we discuss about these thrust areas, these are the not only the research thrust area we have discussed, and also we are working towards the 12 different center of excellences that we have at JSS AHR innovation or ecosystem. And most of the center of excellences which you are seeing on the screen, they are not only funded by JSS AHR. Most of the research center of excellences are funded, are recognized by major funding agencies. Like maybe when I talk about senatal site for multi-country vaccine safety studies, it has again recognized by WHO. Likewise, we have CMR laboratory at JSS Medical College that is again recognized by various funding agencies. Likewise, we have different funding like a center of excellences. Like say recently, we have a center called Center for Advanced Drug Research and Testing, that is CADRAT. That is again approved by the Drug Controller General of India again, and we have DTL laboratories, and again we have a special center of excellence. Recently, it got opened up, that is Computer Aided Drug Delivery Center of Excellence. That is again we have here. I need to specifically mention that we have procured two major tools for the computer drug delivery suite. That is, one is Schrodinger tool, and one more is molecular solutions. Basically, this molecular solution recently we have purchased. Again, this molecular solutions, the workshops will be conducted. Maybe we are planning, maybe this month again, two workshops are coming up, and maybe next month again in at UT we have planned for the molecular solution workshops. Likewise, whatever the center of excellences we take up, it is not only the government funded, and also we have industry sponsored facilities. Like we have tie up with Philip Healthcare, and we have the collaboration with Colgate, this one Imera, Nova Biomedical. Likewise, we have a Section 8 company called as Sparkle Cine also. Like these are the different things which I wanted to mention about center of excellences. And when it comes to the research infrastructure at JSS AHR, we are towards the strengthening the research infrastructure. Maybe when I talk about research intensive university, like it also covers the university central instrumentation facilities, that is called as USIC facilities. That in detail I'll be speaking for 5 more minutes. And we have a well-established tissue culture facilities at JSS AHR, both at JSS College of Pharmacy, Ooty, and Mysore. Recently, we have opened up a tissue culture, cell culture facility, and also we have tissue cell culture facility at JSS Medical College, and as well as at School of Life Sciences in Mysore campus. And we have AAA accreditations for Animal House facility, and we have conducted a very unique program during the DST Stuti program called as Science on Wheels, wherein in Science on Wheels program we have a specially constructed bus, wherein the bus will move from one area to other area, wherein it showcases the advanced technologies for the school children. Like maybe it is a government school or a private school, wherein we showcase them what facilities or what research can be done in India. Maybe it is a kind of creating awareness from at the student end. Again, we are more towards the translational research. Like whatever the research we take up, it is towards the translating the research. And as I mentioned earlier, we have a specific instrumentation facilities called as University Sophisticated Instrumentation Center, that is situated or based at JSS College of Pharmacy, Mysuru campus, wherein we have many instruments were placed. But when it comes to the objectives of the USIC facilities, this is not only the facility created for the in-house research community. Again, this is again the door is completely opened for the even for the external research collaborators. This is what I wanted to bring it in this platform, because most of the participants I can see throughout India. That's why you can utilize our facilities, and we are the registered USIC facilities registered under ISTEM facility. And like what we have at USIC facilities, I'll be taking through maybe in 2-3 minutes. I'll showcase what facilities we have at USIC Mysore. Maybe when it comes to the advanced research instrumentation at USIC, we have NMR spectrophotometer. That is, we have a high resolution NMR facility at JSSHR Mysore, and we have confocal microscopy facility, wherein we have GC-MSMS facility, and we have advanced flow cytometry is available at USIC. Again, we have LC-MSMS system with SciX triple quad 4500. And we also have HRMS facility, TTOF system with again model number 6600 with SciX model. And again, we have some minor instruments like micro ultracentrifuge and zeta sizer. And as I mentioned earlier, we have a CAD facility. Both we have the Schrodinger packages and also we have molecular solutions. Molecular solutions is a tool wherein we have packages specifically for the pharmacy faculty members, and as well we have the basic packages for the life science faculty members, and whatever the packages required for the medical faculty members, we have procured all the packages. And now the software is in, maybe we have installed everything and we have purchased a CPU, wherein I need to mention a particular thing that the CPU which we have purchased is in, like, one CPU is having 128 systems. We can, I would like to mention that. That is when we discuss about docking and simulation, when it comes to the drug discovery, maybe the time which has taken earlier to dock or simulate, do the molecular dynamics of a particular compound, which used to take us like 4 days or 3 days, depends on the internet speed. But now, basically, when we have these molecular solutions, by having this package within a day or maybe in 5 6 hours, we can finish our job of molecular docking and simulation studies. And maybe again, these facilities can be utilized by the other faculty members by having collaborations with our faculty members at JSSCHR. And we have refrigerated centrifuge facilities at USIC facility. And these are the things which I already have mentioned. And again, when it comes to the of advanced analytical technologies, we have ATR, FTR spectroscopy, Zetasizer, and flow cytometry was there. And recently, we have purchased the sorter also. Advanced cell analysis and sorting technology can be done together. That is the make of this model is BD Sciences, wherein we can do deep immunotyping assays and cell cycle analysis of different cell lines can be done, wherein we can connect this all these things into drug discovery. This is what I wanted to tell. And again, we have advanced biotechnological tools like equipments like 3D bioprinter, wherein this 3D bioprinter is the make of this 3D bioprinter is again, this is procured from Cell-Link. And the major equipment, earlier the BioX Cell Link was placed at IASC, and many of our faculty members and researchers, they went to IASC and they have learned how to develop the biofabricated transplantable organs by using this 3D bioprinter. And later we have purchased this BioX, that is 3D bioprinter. And now at USIC facilities, we have placed this 3D bioprinter, right? And we have a next generation sequences facility available at both at JSS Medical College. Like in JSS Medical College, you can utilize this facility. And what we do by using this NGS, I'll tell. We have a specialized laboratory called as genome lab, which is there at JSS Medical College at Biochemistry Department, wherein we have Ion Chef and Green Studio SS plus sequencer, wherein we have the sequence studio Sanger sequencer. Like what we can do by using this, utilizing this genome lab facility is we can do the sequencing services. We can avail, or you can avail the sequencing services for the Sanger sequencing. And if you have anything which is related for the whole exome sequencing, you can please connect to us. And whole transcriptome sequence can be done by using this sequence services. And microbiome sequence can be done, and 16S rRNA metagenomics can be done, and phylogenetic analysis and functional annotation can be conducted by collaborating with this genomic laboratory facility. And again, as I mentioned earlier, we have a facility at USIC where you can book your facilities by going into the website, wherein you have a facility called request a facility or book your slots. Like by going to this either through USIC, you can go to JSS AHR website, you can book your slots. Otherwise, also you can go to the iSTEM portal, wherein in iSTEM portal, whatever the minor instruments we have, even that also we have incorporated into the iSTEM system. That is, iSTEM system is a system wherein Indian Science, Technology and Engineering Department facilities have mapped all the equipments throughout India. Wherever the researchers are sitting, they can utilize the facilities which is available through the iSTEM portal. Again, we have linked our facilities, whatever the facilities we have at JSSCHR, and we have linked the whatever the equipments we have to the iSTEM portal. What I request all the participants, to connect to the iSTEM portal, see to the instrument which we have at JSSCHR, and you can book your slots through iSTEM portal. And we have started receiving many of the samples from Northeast region or North region or South region. From many of the state universities, we have started getting the equipments for the this one. With this, I'll end my institute, whatever the advanced research facilities we have at our institute. And from now onwards, I'll be taking through today's session, how we have divided, my team have divided is first, basically, I'll be talking about what are the useful things which you need to remember when it comes to the clinical trial design and drug repurposing when it comes to the by using the bioinformatics techniques. And after this, what we have planned is take you through the demonstration process. Just give me a second. Yeah, sorry for the inconvenience. Yeah. Maybe now what I'll do is first I'll discuss completely about how the clinical trial design and drug repurposing by using bioinformatic techniques. What are the different things we need to take it through the review of literature, or maybe when by reading the different research articles. I'll just take you through the things which needs to be remembered when it comes to the clinical trial design and drug repurposing. Later, what our team will do is they have arranged for the demonstration wherein they will take you through how the drug repurposing can be done and how the easily the docking can be done. What are the different things to be done before we start the dynamic studies. And when it comes to the pharmacology, this one, everything will be demonstrated by our team, wherein we have today in our team. Yes, please.
Completed major experiment. All of you.
I request
Very
Maria ma'am
few
to
students
mute
have
your
showed
please.
no.
I kindly request Maria madam to unmute. Sorry, mute.
Huh? What about the last?
Hello, Maria ma'am. Ma'am, can you mute your audio? Yes, sir. Yes. Please, sir.
Yes, ma'am. Yes. Yeah, I have with me today, I have Dr. Bhargav Srivatsa, and he'll be showing us through one package. And maybe after that, we have Dr. Anisha Jain, and she'll be talking more about drug repurposing. And then we have a faculty called Dr. Chandan D, and he will take you through how the mapping will be done and epitope mapping will be done, and how the immunoinformatics will work. These are the different concepts what we have planned for today's session. One by one we will take. Maybe I have I have got my time. Maybe I will take some 20 minutes to complete my slides. I'll request Dr. Bhargav to take through his demonstration, whatever he has planned. After that, I request Dr. Anisha and then to Dr. Chandandi. This is how we have planned. And whatever the question and answer questions or any queries you have, you can just drop in the chat box. Later at the end of the session, we can take up the question and answer sessions, right? With this, I'll start my thing, that is in this slide. Again, at JSSCHR, we have a facility called as Scopus AI. Scopus AI is a package wherein when we give one word or one keyword, it gives a concept map. By using that Scopus, here you can observe, by using the Scopus AI, what I have done is I have taken a keyword called as clinical trial design and drug repurposing. When I gave the two keywords, it has given me a nice concept map, wherein in this slide we can see how AI can be used in different things. Maybe when I discuss about drug repurposing strategies, what are the different things needs to be learned. And when it comes to the bioinformatics techniques, what are the different techniques I need to learn before I start my whatever the multi-omics things which I am planning to integrate into my studies. And then it comes AI approach. What different AI approaches needs to be adopted when it comes to a research. Maybe when you are writing any research articles, or maybe you are writing a project proposal for the different funding agencies, how it is useful, let us see. Say, for example, this provides in this slide, what it provides is this is an overview of how bioinformatics and artificial intelligence are transforming the clinical trial design and drug repurposing. This is what the main intention of creating this slide. At the center, what you can see is the different things which can be which you need we all should learn. It is a kind of framework which we need to remember. When it comes to the drug repurposing strategies, maybe the drug repurposing means maybe finding a new therapeutic uses for the existing drugs which is already available in the market, instead of developing a completely new drug, which may take definitely it will definitely take some 10 to 15 years. And probably the coming to the costing of the material, billions of dollars needs to be put in to identify the approved drugs that may work for the other diseases. And when we take up this drug repurposing strategies, we need to remember the drug repositioning, and other thing we need to remember is phenotypic screening. When it comes to the drug repurposing, what it does actually, when we think about the drug repositioning or drug repurposing, this identifies the new indications for the existing drugs. Like the best example is during COVID-19, you only have seen how the repurposing of the drug have taken place. And when it comes to the phenotypic screening, what happens? This observes how drug affects the cell, and maybe how it affects the tissues, or maybe how it affects the organisms. These are the different things we need to remember. And ultimately, what it does is it helps to discover the unexpected therapeutic benefits can be studied through these two things.
Program. My
Then
OT suggests collection of
maybe Jubi ma'am, what we can do is maybe we can unmute everyone, ma'am. Maybe if that option is available, we can mute everyone, and maybe yeah, that option we need to remove, ma'am.
यो करा अगले महीने करा
Maybe Ramesh sir is speaking now. That's why what we can do is, Jubi ma'am, are you there? Jubi ma'am, anyone there from JSS College of Pharmacy UT?
Sir.
Jubi ma'am, what
Yes,
we
sir.
can do, ma'am, you can tell the IT people to mute everyone, ma'am, because
Yeah. I will only do it, sir. No problem.
it can be done, ma'am, from your end.
Yeah. I'll only do it. No
That's
problem.
great, ma'am.
Yeah.
That's great. No?
One
Maybe
minute,
whenever,
sir. 1 minute. I'll first mute all, then I'll unmute you.
Sure, ma'am. Sure.
Ah.
It can be done, ma'am. I'll wait.
Sir, you can.
I can, no, ma'am, no?
Yes.
Yeah, I was talking about the phenotypic screening. And this approach, exactly like I was telling, during the COVID-19, we have taken up several approved drugs, and like we have again rapidly we have evaluated for their whatever the new users were there. And when it comes to the bioinformatics techniques, like bioinformatics provides the data-driven foundation for the modern drug discovery. Like when it comes to the techniques, we can divide them into two major components here. One is multi-omics integration, wherein in the multi-omics combinations or integration, we can combine either genomics can be combined with transcriptomics, or maybe we can combine with the proteomics and metabolomics. Here I would like to mention my specialization is like proteomics and genomics. I work on a particular microorganism called as leptospirosis. Leptospirosis was my interest, and through that only I have got the DST grant, and I have worked on especially on this genomics and proteomics data. Anyone in this group would like to collaborate for any of the genomics, or maybe when it comes to the multi-omics integration, if anyone are looking for the collaborations, definitely I am open for the any kind of collaborations from any institutes. At the end, I'll be sharing my contact details with my email ID. Anybody, any professors can be connected to me for any kind of assistance from my team. And when it comes to the, yeah, we will go for the next thing, that is data analysis. When it comes to the data analysis, what happens? like the whatever the extracts meaningful information from a large, or maybe when it comes to the complex biological databases, when we do the analysis by using the big data, then it comes to the data analysis. Definitely this data analysis supports the patient, maybe we can say the stratification, or maybe like nowadays we are more discussing more about the precision medicine, and these approaches can be done by using this data analytics. And in simple terms, what I would like to say is bioinformatics helps us like to convert the biological data into the actionable knowledge. This is what like when we have this bioinformatics techniques in our hand, definitely the multiomics integration and data analysis would be very easy to do. And ultimately, when we club this drug repurposing and bioinformatics techniques by using the AI approaches, what happens is this artificial intelligence will add the the predictive power. We can say that the predictive power to the processes. Like here, the two major components for this AI approach is you need to know more about the deep learning things, that is DL, and also you need to know more about the machine learning things. When it comes to the machine learning concepts, here when we learn the patient responses, or maybe when we recruit the successfully the patients, or when we do the potential drug candidates identification, then definitely our machine learning concepts will definitely support other two components, that is drug repurposing and bioinformatics techniques. Ultimately, what happens? The AI components will ultimately help the researchers to make the research very faster, and it will be more informed. Maybe the decisions can be taken in prior. And like how these three areas work together when it comes to the real strength, like when the real strength comes from the when we integrate these three components, that is the when we inculcate this bioinformatics with AI component, with whatever the adopted strategies for the drug repurposing, when we do all these three things, then only we can achieve the clinical trial design, and we can ultimately achieve the international standard. We can align to the international standard alignment. Like at the bottom of the thing which I have given is we can also align with the we have whatever the international standard alignments are there, maybe ISO 21015 or 15289. For all these things we can align. These standards ensure that the research remain reliable here, and research remain reproducible here, and whatever the research we do globally, it should be acceptable. Then only we can say the research is successful. The key whatever the take home message what we have is modern clinical trial designs is no longer driven by sorry no longer driven by the laboratory experiments. By combining the bioinformatics, multiomics data, like artificial intelligence and drug repurposing strategies, we can definitely accelerate the drug discovery, and we can improve the whatever the patient selection I have mentioned here, and we can also reduce the cost of the experiments, and ultimately we can deliver better treatment to the patients which are in need. That is what ultimately what we need, as well as from the pharmacy research, as well as from the life science research. What we need ultimately is to serve the patient's need, what the patient is requiring. And these are the different things which we can do when we inculcate all these three things. And then coming to the next slide, what I have planned is, again, I have not wasted my much time in preparing the slides. What I have done is I have collected the research articles again through Scopus AI tool. Again, whenever I give the keywords to the Scopus AI tool, it gives me the idea where and all the challenges are there, wherever where we are lacking with the facilities, or where the training is lacking. These are the different things can be known when you use these kind of tools. We are fortunate enough from our leadership, we have got this tool, that is Scopus AI. If any faculty from JSS AHR attending this workshops, definitely please, if you are not using the Scopus tools, please start using this tool, that is Scopus, whatever the tools which we have procured exclusively for the JSS AHR users. or for the outsiders or for the attendees, those who are from the other institutes also can collaborate to know more about this Scopus AI tool. And coming to this slide, that is multiomics and artificial intelligence. Here, let's walk through. Let us do the walkthrough from left to the right of the slide. Wherein, on the left side, the major things what we can see is the traditional drug development. That is extremely whatever the high risk what we have. More than 90% of the drug candidates fail during the clinical trials, despite promising laboratory results. Many drugs do not show sufficient safety, or maybe when it comes to the efficacy or to the humans, it is not there. Like completely 100%, this is not there now. What limitations of traditional models what we face is historically when we see the drug discovery followed a one drug, like one target approach was there earlier. There is one drug will be there for one target, but that is not there now. Like we need to adapt many of the drugs to do the repurposing now. However, most diseases, what happens here is most diseases are much more complex and involve multiple genes, multiple proteins are involved, and multiple pathways are involved, and maybe the environmental factors also affecting here. These are the different limitations of the traditional things when we adapt. Maybe when it comes to the inadequate, in some of the laboratories there may not be much of the cell line facilities available, or many of the facilities like animal models facility is not available. Maybe basic organoid, like nowadays you wherever the research is happening, we can hear a word called as organoids or spheroids. These are the different techniques are not available. Often we when we don't have these things, then often we fail to fully mimic the human disease biology. That's why the result is high clinical trial failure rates we can face, and maybe the resources can be wasted during this thing, or maybe delayed access to life saving therapeutics definitely we may face when we adopt these things. When we understand the disease complexity by adopting different omics technology, that is as I mentioned in the earlier slide, when we integrate the multi-omics with AI in human relevant models, enables the system level understanding of the disease. Wherein when we adopt the genomic things, definitely we can study the DNA in detail, or maybe the genetic variations can be dealt in detail, or maybe the epigenomics examinations can be done, like how the genes will be switched on and how the genes will be switched off. These are the different things can be carried out by adapting this epigenomics things. Or maybe when it comes to the transcriptomic thing, what we can do is we can measure the gene expression, or maybe if you are learning any RNA activities, definitely when we adopt this transcriptomics into your research, definitely more about the RNA and gene expression can be learned. When it comes to the proteomics, we can analyze in detail about the proteins which performs most of the biological functions, which can be dealt through adopting the proteomics technologies. Or maybe studies which is related to metabolites and the biochemical pathways. Like each layer provides a different perspective of diseases. Maybe we can also think about the complexity of the diseases, where we can think about understanding the DNA. Ultimately, the DNA is a blueprint, wherein RNA is a instruction manual, wherein the proteins are the workers. When it comes to the metabolites are the final products and activities, which when we combine all these things, they provide the comprehensive view of the complete disease mechanism. And what is the role of artificial intelligence here? When it comes to the artificial intelligence, what happens at the maybe at the center you have learned how the AI can be clubbed and how the different omics can be clubbed. And what happens when we do all these things is from data to biological insights can be learned. That is what the key takeaway of this slide, that the future of the drug discovery lies not in reducing the complexity, but in understanding it through the convergence of multiomics and AI and human biology. This is the smarter way of learning the therapeutic uses, And this is how the multiomics and artificial intelligence can be clubbed together. The overall, like again, the key take home message for this slide is the future of the drug discovery is not about studying one gene or one target at a time. It is about understanding the disease as complex biological system, and maybe by integrating all these multiomics technologies, or maybe artificial intelligence and human relevant models, when we can accelerate the precision medicine and deliver it safer in the safer mode, or maybe the effective therapeutics can be delivered to the patients faster than ever before, can be done when we adopt this multi-omics technology and artificial intelligence for the precision drug discovery and potential clinical applications. This is all about this slide. Probably when we take up the AI in clinical trial design, maximum like this slide demonstrate how the artificial intelligence is transforming the clinical trial design by making the trial smarter and faster, and again more efficient, as I said earlier. Like traditionally, the clinical trial, when it comes to the clinical trials, they are very expensive when we adopt the clinical trial. And maybe when we discuss about the clinical trial, again, it is a very time consuming, often faces challenges such as maybe when it comes to the patient selection, or maybe the protocol, like when we design any clinical trial, we need to design the protocols. Always we should have a proper protocols, otherwise if any inefficiencies in the protocols, definitely the clinical trial is going to be a big failure. Like high failure rates will be there. And when we adopt this AI into this clinical trial, what happens is it helps the researcher to address these challenges in multiple stages, not only in one stage, in multiple stages, the throughout the clinical trial process. When we adopt this AI, definitely it becomes a huge benefit for us. Maybe when we discuss about like patient selection and recruitment, what happens, the first application of AI is this only, like that is patient selection and recruitment. One of the biggest challenge in clinical trial is finding the right patients, like who meets the eligibility criteria, or when we adopt this AI, what happens when we adopt this EHR system? EHR, everyone knows, that is electronic health records. When we adopt this electronic health records, definitely like analyzing the patient data becomes very easy. And when we have a data of genomic data banks, or maybe whatever the omics technology what I have mentioned, when we have the large data sets, definitely the training can be done for the AI tool. When we train the AI tool, these analysis data analytics will become a very easy thing. And also the clinical history, when you know the clinical history, this is again a big thing which when we adopt the patient selection and recruitment. And when it comes to the patient selection also, we need to consider the demographic information to identify these patients who are most likely to benefit from a treatment and successfully completing this study. Then only it becomes a complete clinical trial. To this improves the recruitment efficiency, and also helps enroll the right patient at the right time. This is what when it comes to the clinical trial, everyone needs. Then what it comes when we tell about trial optimization, the second area of clinical trial design is the trial optimization. What do you mean by trial optimization? And when AI component is inculcated into this, what happens? That AI can predict what treatment response the patients are giving, and ultimately we need to design what is the optimal dosage of the drug whatever we are using for the clinical trial, and what potential risk the maybe the adverse effect the patient will be having. These are the different things we need to remember. But when we inculcate, or when we involve this AI component into our clinical trial, what happens is this trial optimization will become very easy and very easy process. This allows the researchers to know or to reduce the unnecessary, maybe when you are unnecessarily the study arms whatever you are planning can be limited, and maybe the sample size can be optimized, or you can short down or cut down the trial durations can be cut down by when you adopt this AI into the trial optimization. What do you mean by then virtual trials? When we adopt this AI-enabled virtual or decentralized clinical trials, when we have, instead of requiring patients to visit the hospital frequently, the data can be collected remotely, maybe through the wearable devices. Nowadays, everyone have the awareness about the wearable devices, or maybe through some mobile applications, the data can be collected, or maybe the remote monitoring systems. This is again one of the upcoming area of research, that is how patients' data can be collected remotely, or remote monitoring systems, how the data can be collected. And definitely what happens, this will definitely improve the patient convenience, and this will also increase the participation of the like whatever the recruited patients are there, actively they participate in the clinical trial. And ultimately what happens, this definitely reduces the monitoring whatever the systems we are monitoring. These are the different things which you need to remember in these 3 points. And when it comes to the predictive analysis, the fourth component of the AI component that can analyze the trial success probabilities, or maybe when it comes to the recruitment performances or clinical outcomes. Definitely these things, definitely the sponsors also we need to think. This helps the sponsor to make better decisions before they invest in significant clinical trials. In simple terms, what AI can help is to anticipate the problems before they occur. That is what the predictive analysis can be done for the better outcomes. What better outcomes can be expected when we have this AI better decision, better healthcare? That is definitely the AI component insights. It will give the insight to the researchers in a make a smarter decisions, maybe to improve the clinical trial success rates, or to reduce the developmental whatever the timelines which the researchers have kept. This ultimately benefits both the healthcare systems and as well as for the patients. But still, after adopting all these things, still there the challenges still remains. That is all about the data quality. AI is the only good the thing which when we adopt, this maintains the good data where it receives, or whatever the incomplete data is there, it identifies early only, and whatever the inaccurate results are there, it identifies early, and the biased data will be taken out when we adopt these AI tools. But still, the when we adopt this AI, still we face the challenges. Ultimately, we should train the AI model. Without that, the success rate we cannot find. And the one more major thing which we can observe is ethical consideration. We must ensure that like the patient privacy should be maintained, and the fairness should be maintained, and the transparency of the patient should be kept very like promptly, and responsibility of usage of AI should be thought thoroughly to the users. And AI tools must comply with the regulatory whatever the standards compliances we should have in our mind. That is building trust in AI is just as important as developing the technology for itself. That is like a key take home message for this
അനഘ
slide.
നസ്രിയ റാനിയ ആരാധന മാഡത്തിന്റെ എക്സാം എഴുതണമെന്ന് പറഞ്ഞായിരുന്നേ 2 മണിക്ക് തന്നെ. ആരാധന മാഡം എക്സാം എഴുതണമെന്ന് പറഞ്ഞു 2 മണിക്ക്.
Lakshmi madam, I muted him. Yes, sir, you can. Sorry, sir, you can go.
Problem, ma'am. No problem, ma'am. Maybe a few of the faculties are working and they are listening, ma'am. That's why this
Yes.
confusion is not a problem, ma'am.
Okay.
Like when it like the next thing which I would like to mention, maybe after this slide, I may hand over the session to Dr. Bhargav. I'll just maybe I can take some 5-10 minutes more, and then I'll hand over the session to the demonstration. And here in this slide, what happens is this slide illustrates how artificial intelligence is revolutionizing the drug repurposing, which means how the funding for the new therapeutics uses for the already existing drugs. Traditionally, when we develop a new drug, again, as I mentioned earlier, it will definitely take some 10 years and cost billions of dollars. drug repurposing offers a faster alternative thing by identifying the approved or previously tested drugs that may be effective against the different diseases. When it comes to the computational screening, what happens? The first application is computational screening. Here, AI analyzes the vast amount of biological and chemical information which is already there in the databases. Maybe to identify the drug target interactions, or maybe to identify the molecular structure of any protein which you are looking, or any biological pathways if you are looking. Already the databases are there. Maybe if you are looking for any disease mechanism system, if you are looking using, maybe by using the machine learning, that is ML, or by using the network based approaches, this AI in drug repurposing can definitely rapidly it can identify the drugs that may be effective against the new diseases. Instead of testing the thousands of compounds experimentally, the researchers can prioritize the most promising candidates by using the in silico, by adapting the in silico methods, saving definitely it saves a lot of time, and maybe we can say it saves a lot of resources also when we adopt this computational screening. When it comes to what cost can be cut down, or maybe how effectively the time efficiency can be improved when we adopt this AI in drug repurposing, is one of the biggest advantage of AI driven drug repurposing is definitely the existing data, such as we have a huge genomic data facilities available. Already the database is there. Already we have a lot of databases available for the proteomic data, or maybe the clinical trial data is also available, or the real world patient data to identify all these potential therapeutic applications when we adopt. Because what happens is, safety information for many approved drugs already it is exists. Researchers, what they can do is they can often move directly into the advanced clinical evaluation, and ultimately the significantly what happens is it reduces the development time cost and time efficiency. Applications in what happens when it in the applications of global health, AI powered drug repurposing has demonstrated the tremendous value during the public health emergencies. Well-known example, as I mentioned, during the COVID-19 pandemic, where AI tools were used rapidly to screen the existing drugs for the potential antiviral activities. This approach, what happens is this approach have allowed the healthcare systems to respond more quickly to the emerging diseases, and whatever the urgent medical needs were there, it has been addressed during this global health problems. And again, the precision medicine, like major funding agencies are concentrating on the precision medicine. When we adopt this AI enabled precision medicine systems, like this is again another very important component we should see is another important benefit is contribution to the precision medicine. What happens is not all patients responds to the treatment in the same way, right? When we inculcate, or when we adopt, or when we integrate this AI into the system, then the clinical, based on the clinical information what we have, and the genomic profiles what we have, whatever we have obtained from the patient side, and by having the biomarker data with us, to identify which patients are most likely to benefit from a particular drug. This helps the clinicians to select the right treatment at the right time for the right patient. This is what the precision medicine, when the AI is enabled. Like key benefits of all these things is data, like this will be data rich insights, and ultimately to identify the new users of the drugs, and for the faster development of the drugs with a lower cost. And the challenges what we face, again I would like to mention here, is we should have a quality data, and we should have a built model which is already trained. AI model should be there, where it acts as a black boxes, making the results hard to interpret. And maybe the validation part should be taken care, and keeping the regulatory and ethical things in our mind. With this I'll end my session. Like the summary of the slide for this things, everything I'll be sharing with Anand sir. Maybe after the session, if time permits, again I will come back and I have few more points to tell. Maybe I can discuss if time permits. Already I have taken 1 hour. Maybe I'll now I request maybe Dr. Bhargav or Dr. Anisha. Like
Hello,
just
sir.
yeah,
Yeah.
Bhargav.
Yes, sir.
Yeah. Okay. Then I request Dr. Bhargav to take care of his demonstration. Maybe after that, Bhargav, you can
Hello.
the
Am
next.
I audible, sir?
Yeah. Yeah. Audible, Bhargav. You are audible.
Yes, sir.
Now I would like to mention a few points about Dr. Bhargav before starting his presentation. Dr. Bhargav is he has done his PhD from JSS Academy of Higher Education and Research from the Department of Biochemistry. His PG degree was biochemistry, and his PhD was he has taken biochemistry and bioinformatics and biotechnology as he has integrated all these things. And basically, I would like to mention here is he was one of the student who had obtained the JSSAHR Overseas Research Fellowship, wherein JSSAHR Overseas Research Fellowship, again, every year we have earmarked some of the budget to send our students, those who are in the second year of PhD, pursuing PhD degree at JSSAHR, they have to search for their mentors outside India, and the JSSAHR will support for their accommodation and for their travel and for other things which we will support from JSSAHR. Only thing, they need to search for their team leads. As Dr. Bhargav in the year 2022, he went to University of South Carolina, USA, United States of America. He was there for almost for 8 to 9 months. He learned many things, like maybe in his slides he'll be covering how a conventional drug can be taken up, and how from the phytochemicals the components can be how a library can be created, and how this library, how he can adapt those libraries and screening the larger databases, how it can be narrowed down, and for the particular diseases, how this all, maybe if you have a 1 lakh library of compounds, how this 1 lakh library of compounds can be utilized to identify targets for the different diseases. These are the different topics which he learned during his stay at South Carolina University. Likewise, we have another candidate after this session, that is Dr. Anisha, and after that again we have Dr. Chandan. They are unique in their techniques. Now I request Dr. Bhargav. Now he is working in the Department of Microbiology, and he is into completely his research is into drug discovery. I request Dr. Bhargav to give his inputs and give his demonstration related to the drug discovery and diagnostic approaches. Thank you, Dr. Bhargav, for your time and for giving this demonstration. Yeah, over to you, Dr.
Thank you,
Bhargav.
sir. Thank you for the introduction, sir. So, hope I'm audible and my screen is visible. Hello, sir. Am I audible?
Yes, yes, Dr. Bhargav, you are audible.
Yes, sir. Thank you. Thank you, sir. Thank you for the introduction. Good morning. Good afternoon, all. I hope Dr. Chandan, sir, has already covered the most of the parts of the artificial intelligence in bioinformatics and drug discovery approaches. As we all know that we have a conventional drug discovery pipelines that includes starting from the bioinformatics, from screening down the larger molecules to the lead identification and optimization, which further carried by the preclinical studies and the clinical trials, and ultimately ends with the regulatory approval for the drug marketing. So there are a lot of challenges when it comes to the conventional approaches. The main thing is all about the time consuming and expensive at the same time, and there are a lot of high rate of failures when it comes to the new drug candidates identification, and they requires a lot of extensive laboratory and infrastructure for the development of the new drug molecules, and the diagnostic variability as well. So most of the drugs, whatever that has been manufactured or discovered right now, is not a personalized medicine or a precision medicine kind of one. So most of the drugs, whatever is discovered, is keeping in the mind of the Western population, and not the South Asian or the Indian population. This population-specific medication is much required when it comes to the new drug discovery approaches. And coming to the emergency of the artificial intelligence in the bio mainly focused on storing and processing and analyzing the biological data. However, the rapid growth of the high throughput technologies has led to an unpredicted exploration of the biological informations. So now, whatever the drug discovery or the disease diagnosis is occurring with the usage of the artificial intelligence, this is going to be an breakthrough where it goes in deeper into the biological data and understand the biological data, and then it is going to interpret the biological data. It is not as the conventional bioinformatics approaches. The modern research now generates vast amount of the genomic sequences. Like we have the genomic sequences, we have the transcriptomic sequences, proteomics data, metabolic data, and we do have the structural biological data. Maybe when it comes to 3D structural protein structures. And we do have the imaging data, which has to be taken into serious consideration for the disease diagnosis, because most of the drug discovery starts with the disease diagnosis and identification of the targets, and then starts with like we do have the electronic health records, which is very much required when it comes to the personalized medicine and the treatment approaches. Analyzing these diverse data using conventional statistical methods has become increasingly challenging dimensionality and heterogeneous nature. This is where the artificial intelligence has emerged as a transformative technology. The AI enables computers to identify hidden patterns. So basically, it is all about when it comes to the imaging, we are going to make them as a small vocals. So when it comes to the MRI data or the CT scan data or the PET scan data, as we all know that there is a pixel for the regular images, and this pixel is the simplest form of the image. In a similar way, we do have for the MRI, which is the tiniest unit called as acceleration for the data identification. This is what that AI enables the computers to identify the hidden patterns of the images, learn from the massive data sets, and makes predictions with minimal human interventions. By integrating, and one more thing is all about we do have a lot of AI models for the drug discovery approaches, for the disease diagnosis and early predictions of the diseases as well, where the accuracy rate of the artificial AI model or ML or the deep learning models is which is limited because they have a single data set which is utilized for the predictive data, predictive model development. So our team basically we work on the multimodal data integration, where we use just the genomic data or the transcriptomic data along with the imaging data and the electronic health records. So we integrate all these three different data sets and build a and we are going to build a combinational a predictive model for the development of the predictive tools. So basically, I'll just we do have when it comes to the target identification, we have a disease gene prediction tools, which where the AI tools is going to utilize for the identification of the when it comes to the Parkinson's. Let us take an example of a Parkinson's disease or Parkinson's disease. So there are a lot of AI tools just by giving a name of a disease condition and the classification whether the disease condition is in the class 1, class 2, or stage 1, stage 2. If you specify that the disease gene predictions can be done using that AI applications. And when it comes to the structure predictions, we use a lot of structure predictive tools like we have BioVR Discovery Studio, we have PyMol for the structural predictions, and we do have Swiss model for the structural predictions. And we have artificial intelligence application where it can identify the misfolding of the protein as well. So it is going to identify it's going to detect the sequence of the protein. It is going to build a structure and it is going to identify if there is any misfolding in the protein of the structure. Whenever there is, we all know that the protein misfolding is the major problem when it comes to the most of the mutations or the disease conditions which is occurring. And when we have a lot of more number of molecules to be screened for the drug discovery approaches, we go with a virtual screening method, where we can screen lakhs of molecules and we can take them to the pick up more 5 or 10 potential molecules for the in vitro and in vivo approaches for the drug discovery process. And we will have a lead optimization where we can see the molecular property predictions of the things, And for the toxicity predictions, we have ADMET, and clinical trials, we go the patent satisfaction, and drug repurposing is one thing which can be utilized nowadays for to reduce the timeline for the new drug discoveries. And so when it comes to integrated AI bioinformatics workflow, we have to integrate the different patterns of the biological data. This biological data includes again the multi-omic data, imaging data, and the clinical records, where once the data is collected and it has to be pre-processed before going for the model development or a predictive tools development. So basically what we do when we have a huge data sets, like when it comes to genomic data, it will be in a textual data format. When you have the imaging data, we have now vocal formats, and we have the clinical records as well. So we have to pre-process the data. So by we have to go for the QC, the quality check, whether the data quality is good enough to go for the further steps or not. And then we have to go for the cleaning process of the data, and then go with the alignment of the data and see whatever the missing data information is there. We have to rectify that missing part, and we have to fix them and go for the feature extraction. And once this bioinformatics pre-processing is done, then we go for the development of a model. So there are two components again, as we all know, that is one is with the training and the testing models. So when the model is trained with enough amount of the data, so the data requires large amount of data, when it is tested, and then we go for the prediction and the pattern discovery approaches. So then comes the knowledge discovery, where out of this predictive tools, we are going to identify the new biomarkers or the hypothesis for the disease prediction will be given, and which will be beneficial for the drug discovery and the clinical decision making as well. So now we have a lot of AI tools for the bioinform AI tools used in the bioinformatics and the drug discovery approaches is all about the, when it comes to the protein structure predictions. Basically what we do with the protein structure predictions, we go with the Swiss model or with the PDB kind of thing. But now we have a AI tools where we have the alpha tool and the rostaf 4. So alpha tool is the one thing where it is going to predict the three-dimensional structure of a protein which is not present in the PDB, which is going to be a novel one. So we might have identified any new protein. So we have a sequence of that particular protein, and we don't have any leads for the identification of the structure. In such cases, we can use the alpha fold, so which is going to identify the novel protein structure prediction, which is not present in the PDB or things, and it is proposed by Google. And the second thing is what we have is all about the drug designing. So we have to design your molecules. Basically, in the conventional method, we used to go with a chem sketch, basically for the designing of the drug. And now we have the artificial intelligence tools where we have deep learning framework for the drug discovery and molecular modeling, and AtomNet, MolPal, and Renewvine. So these are the tools which we can use for the drug designing or the ligand designing things. And we have for the drug repurposing techniques. Conventionally, we used to go with databases like Zinc and Drug Bank. All these things are PubChem for the collection of the molecules, and we have to screen and do the lot of literature review has to be done for the going before for the drug repurposing purpose. So now we have a Panda Omics and all other tools for the drug repurposing purpose, and molecular docking and QSR analysis. As we all are aware that JSSA HER has a Schrodinger tool for the drug discovery approaches like molecular docking and dynamic simulation approaches. And there are a lot of other AI tools, that is DeepNock and ChemProp and Morbid. So these tools are helping us with the seamlessly we can do the molecular docking and with the highest accuracy rate as well. And now we have the diagnostic AI tools where, just to give an example, the Apollo Hospital in the Mysore has integrated the AI in the colonoscopy, where this particular AI tool is going to differentiate or classify the cyst and the tumors which is present in the colon. Most of the time, the cyst will be considered as a tumor and it will be misdiagnosed. Such cases can be reduced when we have the AI assistance when it comes to the disease diagnostic approaches as well. So this is the workflow of the AI tool. That is number one with the AlphaFold. How we are going to do with the deal up with using AlphaFold, how we can go for the prediction of the new protein structure. So we should have the amino acid sequences basically. So whatever the new protein that you have identified, go for the NMR or any sequencing method. So you get a sequence of the particular protein. So then it will be subjected for the multiple sequence alignment tools. So all these steps, basically what it is integrated in the AlphaFold tool. So initially it has to be done conventionally by each protein structure sequence has to be retrieved and go for the multiple sequence alignment tools, then go for the HCBI for the evolutionary pattern assessment. And now this particular AlphaFold has already integrated all these packages in it. So where the protein sequence, when we have the protein sequence in our hand, it is going to identify go for the multiple sequence alignment to find the homologous sequence which is similar to it, maybe the 99% or the 100% of the sequence, whatever we are going to get, it is going to take that particular sequence and look into the evolutionary information just to see that the patterns and the relationship between these particular proteins, and then go for the transformative neuronal network, which includes most of the mathematical models here for the structure predictions. And we have something called as where the AI tool is going to predict the angle of the amino acid from which angle to which angle the protein has to be interlinked with the peptide bond, and these things has to, or the disulfide bridges where it has to be bounded, how the alpha helix or the beta it has to be folded. All such things will be predicted using design and angle predictions, which is present in the AlphaFold, and then go for the three-dimensional structure development. And it also gives us the confidence score. Based on the confidence score, we can predict that how confident that this particular protein is in this particular structure or not. So this is how the three-dimensional structure of the protein can be designed using the AlphaFold. And then we have the deep chem where it uses where it can be utilized for the ligand preparation or the ligand structure predictions. We have the chemical structures or the spices which is present in it, and we can go for the molecular fingerprinting technique where it converts the structures into the informative fingerprint patterns, and then go for the graph neuronal network things where it captures the structural and relational information of the molecules, whether it is active molecule and whether this particular molecule is soluble in any solvent or non-solubility in any particular solvent, and drug likeliness can be identified. It basically whatever we have the Swiss ADME technique, so it works on the same principle, but it is completely automated and AI assisted technology. And we do have a path AI for the pathological assessment. So we might be having any like histopathological slides or the histopathological images for the examination, and the whole slide image can be uploaded in this particular path AI. And what this path AI is going to do is it is going to segment your images data into small pieces. So this is what we call it as a voxels or the pixels. So these small segments, each and every small segment is going to be assessed in detail, and it is going to extract the features. Feature extraction is nothing but it extracts the relevant features such as what is the structure texture of the particular image, what is the shape of the image, what is the intensity, what is the morphology, what is the color of that particular thing, and what is the morphology of the internal structures of the particular cell or something. So all these things can be done in the feature extraction, and then it go to the deep neuronal network. This is what actually the main thing which is present in this path AI. So where it uses the neuronal network to analyze the features, whatever is identified in the feature extraction, and it is going to predict the whether it is when it comes to the if the histopathological image is from the or basically cancer tissue. This can be classified. This is basically a classification of the images can be done using this path AI, where this helps in the clinical decision making and the support. And these are the this is how what are the advantages of using these particular tools in the drug discovery. Is all about the fastly we can identify the target and we can go for the drug discovery approaches and more accurately as well. And then when the AI analyzes vast biological data to quick identify and prioritize potential drug targets, then we have the reduced drug discovery time, where this particular pipeline is going to reduce the drug discovery time when it comes to the conventional method, and improves the predictive accuracy as well. And then it helps again with the high throughput virtual screening and toxicity prediction, cost reduction. AI in bioinformatics is going to help us with the precision medicine and personalized therapy as well. So this is all about the more like case studies, and the AlphaFold predicted over 200 million protein structures and evolutionary structural biology. And we do have a lot of these AI assisted technologies now. And when it comes to the AI driven bioinformatics and disease predictions, till now we have understood how the AI tools will be utilized for the drug discovery approaches. Now we are going to look at the disease prediction models. So let us assume that we have a DNA, we have a central dogma theory, as we all aware that the DNA will be encoded into the RNA, and then it goes for the protein coding and it releases the protein. And if there is any small changes or the mutations which is occurring in the DNA, which will directly reflect on the RNA and the protein. In a similar way as well, we do have something called as a mother code or a source code, and this particular source code is derived into the bytecode, and it is going to be the execution. Whenever there's a small problem with the source code or into the bytecode, it is going to have directly reflect into the problem with the execution of the model or the development of any particular task. So then we have again the central dogma theory. So we have the DNA. In the DNA, we have the single nucleotide variation, copy number variations, and the single nucleotide polymorphism. And the transcriptomics, we have RNA expression and the RNA structures. And when it comes to the protein, we have the protein expression analysis. And we do have here is like FASTA-Q is the one. So whatever the genomic data or the transcriptomic data or the proteomic data that we are considering, it's a textual data. It has to be converted into the SAM. So SAM is considered as a sequence alignment map, and then we have the BAM, it is known as binary alignment map, where it is going to code numbers. And let us consider that the whole genomic sequence, that whatever we have is the whole book. SNP or the CNVs are considered as a single every nucleotide sequence. Like to consider the genomic data that we have the large set of databases like NCBI, EMBO, EMSO, UCSC. Like we have a lot of data banks where we can collect the data from them. And basically what we work on the Parkinson's disease. I just don't want to go in detail about the Parkinson's disease. It's a neurological disorder and age-related disorders. So our team basically works on the Parkinson's disease and its early predictions. So what we are doing here is all about instead of taking any single set of the data modality, we are going to consider the multiple data modality into consideration for the model development. So basically what happens when it comes to the like AI predictive models, the deep learning models are mostly considered, are highly considered for the imaging data. And in our work, what we basically do is we are going to use the imaging data, we are going to use the genomic and transcriptomic data. At the same point, we are going to use the biochemical data as well for the disease predictions, the early disease predictions. So what we are doing here, instead of studying in a segregated manner, we are integrating them in a single modality. So for this, we have used the PPMI is one thing that we have the data bank which can provide the data of the Parkinson's disease patients and the control patients as well. So here we can see that we have the T1 white MRI data for the patients of 205, and these particular patients data is now segmentation has to be done once you collect the data. So the MRI data, it is in the imaging data, and this imaging data has to undergo for the each and every small part of the brain or the region will be taken into consideration for the segmentation, so that the model can go in deeper and study about the structural modality or the structural dysfunction in the particular data segmentation. So once the data segmentation is done, we go for the normalization and then go for the image quality control. So then we go for the region of interest, where what is the region of interest, what is our region of interest instead of whole brain. If I consider the Parkinson's disease occurs only, the neuronal loss occurs only at the substantial nigra region. So we consider the R region of interest for the definition and the feature extraction of the data. So where it is going to generate a neuro imaging data. And now at the same point, parallelly, we are going to develop, this is using a deep learning model, and we go for the machine learning model again for the genotyping data. So we use the single nucleotide polymorphism data here, and we go for the sample QC, that is quality control analysis. Then we go for the identification of the minor allele. So it's not about going and considering the whole data set here. We are going to take a smaller, even a single allele is going to consider for the how frequently they are occurring in the sequences, and then go for the genotype call rate. And here we are going to apply a model called as Had-Weinberg equilibrium model. This is actually a mathematical model which will be applied for this, and we are going to integrate the genetic data. So now we are going to merge this genomic data, and here from the imaging data we are going to merge, and we are going to develop a fusion model. So basically what idea is all about here is, so whatever the biological changes which is occurring, or the genomic changes is occurring in the particular region of brain, is suddenly it is going to have an impact on the structural changes as well. So whenever there is a mutation in a particular gene, or the misfolding in a particular protein, and this is directly going to have an implement on the neuronal damage or the structural damage of the brain tissues. So now we are seeing the relationship between this genomic or the any multi-omic data and the MRI data, and we are going to merge them and going to predict the disease early as possible. So this is just a simple method how we are going to analyze it. We have the SNPs and we have the MRI features. Data features is nothing but a region or the texture of the data will be considered, and then we are going to merge them as a multimodal fuse features. Then go for the joint fusion segregation, and we have classification 1 and 2, classifier 2, where we are going to do the testing and the training of the models. So this is how we are going to this is one, the training data representation of the training of the data. So basically there are two data sets which is considered as dual lead and AE stacking. So dual lead is all about we have a single nucleotide polymorphism data which will be taken into local probability model, and it is applied into the global annotation based model. So where it is going to use the same statistical method of random forest and all other probability testing, and it is going to predict the PD early. And now so we have something called as AE stacking. So the dual lead is all about using the single modality data. This AE stacking data, which is considered both the imaging data and the textual data, that is nothing but a genetic data. So the level of fusion that we do, and we are going to predict the Parkinson's disease here. So this is how the MRI and whole genome sequence or genomic sequences will be taken into consideration. The integration of brain MRI and the whole genome sequencing data of comprehensive neurogenomic analysis. So with the same thing, our team has developed an like early detection for the multiple sclerosis using EEG and hybrid deep learning models. So what we do is most of the studies for the multiple sclerosis diagnosing, we consider it as a EEG electroencephalogram will be considered for the disease diagnosing purpose. So what happens is here the healthy neuron is going to give a signal from one, the neuron signaling will be passing from one end of the neuron to the other part of the neuron where the signals will be captured. Whenever there is a damage in the neurons, the electric potential will not be passed from one cell to the one neuron to the another neuron. So these differences in the electric potential will be captured by the electroencephalogram for the identification of the neuronal disease. So here we consider there are something called as in the EEG patterns we have delta power, theta power, alpha power, and alpha peak frequencies will be considered. So when it comes to the multiple sclerosis, there will be an increased level of the delta, and elevated in the theta, and the alpha will be reduced. Alpha values will be reduced. Most of the time, when it goes for the traditional way of disease diagnosing, there are very high chances of misdiagnosing, and there might be very like when it comes to the early predictions, there won't be lot of change with respect to this delta, theta, and alpha power. So patients, when it comes to the early stages like stage 1 or 2 patients. So what we do here, we are going to develop a model, artificial intelligence aided model, so that it can predict or identify the smaller changes in this theta, alpha, beta, and gamma variations. So this particular when we upload our EEG data into the our predictive tool, so this is going to take EEG envelope extraction. So it is going to extract the EEG data from the data, and it is going to classify whether it is an abnormal or a normal cases. So this is just an classification of the EEG data that we have done. And then we have one more model, that is the ANN conjugated thermography model that we have designed. So this particular model is going to, we have a thermal scanner, which is in a helmet concept. So the thermal scanners is going to scan the whole brain, and then it is going to the thermal scanner data will be uploaded to our predictive tool. So where it is going to make again the thermal image classification will be there, segmentation will be done, and then the thermal image acquisition will be done. So the image will be classified and it is going to identify the inflammation assessment and cognition of the artificial intelligence. So this is a brief overview of our model, that whatever we have developed. And so this is how it classifies from the, when we visually see that there is a control group here, there is a disease group here, and this is pattern of second part of the second patient of the disease here. So when we see visually, there is no change in the disease patterns, but when we apply our model, whatever the model that we have built, so when we upload this data to our model, it is going to class control condition or abnormal conditions. And for this particular invention of the systems for detection of neurodegeneration using artificial neuronal networks based thermal office in the year 2026. And we do have one more thing, that is copyright has been registered by the Copyright Office of India for the electroencephalogram analysis. And these are the references for my study that we have considered. And this is all about my presentation.
Yeah, thank you, Dr.
And
Bhargav.
thank you for the opportunity given.
Thank you, Dr. Bhargav.
Yes, sir. Thank you, sir.
Yeah, now, Juby ma'am, if time permits, I'll invite Dr. Anisha now quickly.
Hello, sir. You can proceed, sir.
Yes, ma'am. Thank you, Nivedita. Thank you.
Thank you, sir.
Competition source now to the best profile of this is yes data one paper
It's a visible, ma'am.
now the Q2 Q3
Yeah, I will go there.
and other top sponsor for different data
Slides are visible, ma'am.
Yes, sir. It's visible, sir.
Yes, sir, it's visible.
Thank you, thank you.
Trusted app of the seats plant extract
Very good afternoon to everybody. My name is Anisha Jain and I'm a DBT RA. Pursued my PhD under Dr. Chandan sir. So I'll very briefly tell about drug repurposing in cancer. So this is what I pursued my PhD. What my focus was to identify drugs that can have multiple targets and not just single target. And instead of developing a new whole traditional drug, it's better to find existing drugs that can target multiple mutations at the same time. So I had developed a protocol and a procedure to identify drugs that can target mutations of non-small cell lung cancer. So we have already learned why drug repurposing is important when compared to the traditional drug discovery, when compared to the with respect to cost, timeline, and the approval rates. So there are a lot of articles which confirms that there is an advantageous there's a major advantage of using this drug repurposing concept instead of the traditional drug discovery. So I won't repeat this slide again. So this is what the procedure that I had followed. So what I did, screenshot ಆಗ್ತಿಲ್ವಾ?
Slides are not visible, ma'am.
Just a second. Full. So, ma'am, is it visible now?
Yes, ma'am.
Yes.
Okay, I'm so sorry that it wasn't visible. So here is a workflow that I had followed for my PhD research, wherein I had conducted an in silico analysis, in vitro, and in vivo analysis. So there are various ways of using this drug repurposing concept using an AI or an in silico approach. So one has been already explained by Dr. Bhargav and Dr. Chandan sir as well. How AI can affect and identify various concepts of a therapeutic or a diagnosis basis. Here I have used a structure-based drug repurposing. What this actually means that we are categorizing drugs and we are selecting drugs based on its structure, and targeting also is based on the structure. So we have both the structures of the drugs as well as the targets. So first concept, what we do is we identify the targets that we need to focus on. So since my concept was based on completely on non-small cell lung cancer and specifically on the KRAS pathway. So KRAS is one of the most highly mutant mutation of causing lung cancer. So I focused on choosing the KRAS wild type and the KRAS on and off targets, which is the same KRAS, but it has two different genes. So I focus specifically on the KRAS G12C mutation, and there are several other mutations, but KRAS G12C is highly mutated protein of non-small cell lung cancer. So I selected this KRAS G12C protein along with other co-mutations. So these structures of these targets are already available on the Protein Data Bank website. So Protein Data Bank is one of the most common and highly used repository, which has structures of millions of biomolecules. And if the structures were retrieved from this protein data bank. So if at all the three-dimensional structure of the protein is not available, then we switch to homology modeling. So the homology modeling takes a sequence of the existing structures and it develops a 3D model. So this was done using a Swiss repository, and if the structure is already available, we can easily be retrieved from protein data bank. So next was to identify the drugs. So the drugs were taken from the we can get the individual drug structures from PubChem. So PubChem is one of the repository. We can get three-dimensional structure, two-dimensional structure, and all the related information, chemical information, physical information, pharmacokinetic, pharmacodynamic structures, and all the information of a drug or any small compound is completely available in the website of PubChem. So PDB is for protein structures and PubChem is for the ligand structures. And to retrieve a drug library, so instead of taking individual structures of individual compounds, and if you are focusing on a high throughput virtual screening, then we can take the structures of an entire library. So this library can be retrieved from various number of links. For example, Enamine, Select Chem, and Drug Bank, Zinc. So these are all the websites where you can get the library, entire library, in a single SDF file. So there's just one SDF file that can contain 5,000, 10,000 compounds of structures, and this single file can be used to bind against our specific structures. So we take what we did was we take the structures from the PDB and the ligand structure from Enamine. So we have a library of all the compounds, and whatever the compounds are present in that library can be and all the information is present in an Excel file through the same website. So the further process of preparing these structures of both proteins and ligands were done in the using the Schrodinger website, which is licensed at JSS AHER. So these structures has to be prepared to remove any abnormalities or missing residues or broken chains before we proceed with our in silico analysis. Once this is done, and even there are multiple free software also to do this, but the advantage of using Schrodinger is that everything is in a single workspace. So you don't have to shift to any other profiles or any other software or tools. So everything can be done in just a single workspace. So once we prepare the structures, we then go for molecular docking and molecular dynamic simulation analysis. So what molecular docking exactly does is, I'm sorry, it binds the ligands to the binding site of the target proteins. So there is a binding energy that occurs between the ligand and the binding pocket of the protein molecule, as we know it in the enzymatics of the active site of the enzyme. The similar thing happens even also in molecular docking, where a binding energy score is produced. So based on that score and in comparison with the reference ligand or the existing approved ligand, we screen our compounds based on the energies. So there's something called Blythe score and docking score, wherein we can screen our compounds by setting a cutoff value in comparison with our standard drug. So once this is done, we can further filter our compounds and conduct a molecular dynamic simulation analysis, wherein an in-depth analysis of how our ligand is behaving with that specific protein in a certain period of timeline in presence of solvent molecules, and we can see how stable that ligand behaves throughout that timeline within that binding pocket. This can be analyzed. So this step is very crucial to further screen our compounds for the experimental assays. So instead of taking a lot of compounds to the experimental stage, which is very time consuming and expensive as well, we can directly screen the ligands in these two steps, which is molecular docking and molecular dynamic simulations. So once we conduct this thorough analysis of computational, so we get a lot of data wherein we can filter our compounds by analyzing the intermolecular interactions, the stability, the root mean square deviation graph, and other analysis. And then we take these compounds to cell-based assays. Further, these compounds are tested against various cell-based activities like apoptosis, cell proliferation, cell migration, FACS, and also Western blot to focus on our mutations. So since we have taken certain mutations and compounds, biomolecular structures in silico, so those compounds and those biomarkers can be tested in the Western blot as well as the final step. So further once our compounds are all screened, then we can go for further preclinical and clinical evaluation. So this was the basic another workflow to identify drugs, right from computational screening to final marketing approval. Sir.
Thank you, Anisha. Yeah, thank you. And now I request Dr. Chandan D. Maybe Jubi ma'am, if we have 15 minutes, then we will go ahead. Otherwise, also no problem, ma'am. Already most of the things have been covered by Dr. Anisha and Dr. Bhargav. If time is there, then we will continue, ma'am, the sessions.
You can, sir, no problem.
Okay. Dr. Chandan? Yeah.
Hello? Yes, sir.
Yeah, we have Dr. Chandan D with us, and he is now into Department of Microbiology. Again, he has completed his PhD from JSS. He was actually my PhD scholar, and three of the faculty, those who have presented today, they are all my PhD scholars, and now they are the faculty members of JSS. Now I request Dr. Chandan to give his insights into AI immunoinformatics. Particularly, he is very good in immunoinformatics and in handling pharmacopoeia global and cyber tools. Now I request Dr. Chandan D to give his insights. Over to you, Chandan.
Thank you, sir. So, uh, I think like most of the components, Bhargav and Anisha has covered. So, when it comes for the artificial intelligence, what and all they have talked till now, like predictions of drugs, either it may be repurposed drugs or it may be back to components. Similarly, prediction of proteins or training the AI for integrating and analyzing the novel targeted proteins. So these components, like they have already discussed. So now I just want to talk about one such area like immunoinformatics. So where you can design the peptide or you design the vaccine for the target specific. So that is the one thing. And another thing is I will also be talking about the one of the very useful tool, that is Pharmacodia Global and the Cyber Drug Discovery tool. Let's start with the immunoinformatics. So yeah, the first point I think like Bhargav has already covered, and Anish also talk about this particular component, that is deep learning for target identifications. So when it comes for the immunoinformatics, so let's not talk about in brief about what is antigen, antibody, and all those things. So it's all about the epitopes. So the very first thing when it comes for the immunoinformatics is the very first thing what they will be doing is so using the first parameter, deep learning for target identification. So either from the whole genome sequences or from the NCBI, any of the data bank. If you have the wet lab data, or if you have the or if you are taking the data from the any of the data source, so you will be collecting those data. And also, JoBM has already discussed about the network pharmacology, where you will be identifying the genes and the targeted proteins. So once the genes and protein you have identified and you got to know that, yes, this is a targeted protein. So the next step is using immunoinformatics, how we can design the vaccine, right? So what we'll be doing is, so once you have identified the protein, for that particular protein, you will be looking for the epitopes. I think you can see this yellow color region, that is the epitope. So the surface region where the specific antibody binds to the target. So that is what's called as epitope. So I'll be not talking about much on that. So this particular component, I think like you've already learned this, that is sequence similarity search, multiple sequence alignment, phylogenetic analysis, homology modeling, structural validations. So these are the very basic parameters what you require. So here I've taken an example of leptospirosis, which is one of the major area what my guide and we are working right now on this particular component, that is leptospirosis. So when it comes for the overview of how we'll be designing the vaccine, so the very first thing is, as I told earlier, we'll be taking the sequences, then we'll be going for different analysis like epitope predictions. So again, in the epitope predictions, we'll be identifying the T cell epitopes and the B cell epitopes. Regarding the tools, I'll be explaining in the upcoming slides. So again, in the T cell, MHC class 1 and class 2, then by narrowing the top of, five T cells and the B cells, then we'll be constructing the vaccine. So again, for the construction of vaccine is very much important. So we'll be using the linkers. With the linkers, we'll be also doing some mathematical calculations. So I will not discussing much on the mathematical calculations. I will be just discussing about the linkers, what and all the linkers we'll be using for vaccine constructions. Once the vaccine is constructed, then we'll be going for different analysis to check the toxicity, stability, and we'll be also doing the homology model, and also we'll be predicting structural certain structure prediction thing. Once we have designed the vaccine and once we got to know that yes, this particular vaccine is stable, you can go for two component. One is you can bind any of your drug of interest. For example, like you can conjugate to that particular vaccine your drug of interest. And other thing is like there are different other techniques like um, uh, oligosense nucleotides. So you can bind that particular oligosense nucleotides to this particular vaccine, and you can go for the drug target specific inhibition component. Then we'll be doing a molecular docking studies. Then you'll be going for immunosimulations, and then we'll be doing the MD simulations. So this is the overview of how we'll be validating the constructed protein, constructed peptide till to validation using bioinformatic tools, right? So this is the workflow. again, the same thing. I'll be not repeating this particular component. When it comes for the tools, as I told earlier, IEDB, one of the standard tool what we will be using for predictions, either for B cell epitope predictions. Now, even the T cell epitope predictions can be done using IEDB tool. But now the publications, the researchers are not concentrating much on the IEDB for T cell epitope prediction because of the IC50 value calculations that cannot be directly done using B cell epitope prediction, IEDB tool. So for that, we are using any net MSc PAN 1 and PAN 2 for MSc class 1 and class 2 predictions. So again, will be if you want to understand the population coverage area, like for example, you will be, for example, someone is working on the similar leptospirosis, either from Japan or China somewhere. So if we want to analyze the population coverage area of your target, then that can be done using this population coverage tool. And this is what I was talking about, how we'll be designing the multi-epitope peptide or the vaccine. So we'll be adding the adjuvant. If you have the adjuvant, then we'll be then we'll be adding your B cell epitopes, T cell epitopes, and you'll be having a linkers for each from in between adjuvant to B cell to T cell to your conjugate. So then you'll be doing a second structure predictions, so protein validations and protein, again, the ProStack web tool will be using for protein validations. These are the few results what we got from the basal epitope predictions, MSc class 1 and class 2 predictions. And this is what the results how we'll be getting from population coverage area. This is also has been done using IEDB tool. So, and this is one of the vaccine what we have used in in our research, like we have designed this particular multi-epitope vaccine for sphingomyelin, which is one of the major protein of leptospirosis. In order to inhibit that particular leptospirosis, we have designed this multi-epitope vaccine. And yeah, towards the left, you can see how the vaccine has been constructed. And towards the right, you can see the linkers. And this is what the secondary structure and protein validation has been done. Toxicity, solubility, all the targeted stability analysis has been done. So once that is done, then we'll be understanding how this particular vaccine or how this particular drug peptide, how it is interacting with your targeted proteins. So for that, we'll be doing a molecular docking studies. Then we'll be going for molecular dynamic analysis. After that, we'll also doing the cloning expression studies. When it comes for the wet lab, what we'll be doing is once you have any peptide is designed, you'll be going for cloning with we'll be replicating in any of the E. coli cells. Once you have cloned with that particular component, then we'll be doing the validations, and then we'll be going for the in vitro different confirmatory studies. That is, the vaccine has been cloned to the empty cells proper. Similar, there also in informatics, we'll be doing the cloning analysis. Once we have done with the cloning analysis, there is another component called as immunosimulations, where we'll be checking how the antigen antibody interaction takes place. So this is what we'll be doing with that particular thing. So yeah, I will be not discussing about much on molecular docking and molecular dynamic simulations. So we have used Schrodinger software for molecular docking and molecular dynamic studies. So yeah, this is how the results are. So cloning, how we have cloned with our our vaccine with empty E. coli cell. Then we have done the molecular simulations. So this is how this is molecular docking studies. So how our vaccine is interacted with the targeted protein. And this is the MD simulations. And this is after doing the molecular docking studies and MD simulations, we also did a stability check. So whether the protein and the peptide are stable, whether can interact, and how effectively your vaccine will be taken further. So that we have done using protein peptide stability analysis. Again, these are the results of how we have done the cloning, and this is like how we have done the immunosimulation analysis. So here you can see the different colors, antigen antibody interaction. So there are different colors represents the different component of your analysis. So it's not like only bioinformatic analysis, either it may be any of the tool, whichever you have done. So nowadays the publications are asking for the in vitro validations. So this is what the in vitro validation we have done with ex vivo analysis. So this is a easier model, and we have standardized for different diseases also. So one of the diseases like what we have targeted for anti-leptospiral activity. So we have done the ex vivo cam analysis, and also we have checked with in vivo animal model. So we have targeted for the kidney, as leptospirosis is majorly affecting the kidney. So using a hamster model, we have done the in vivo analysis too for the validation of our vaccine. So how our vaccine has been constructed, whether that is effectively inhibit the particular leptospirosis bacteria or not. If it is inhibiting the leptospira bacteria, how effectively it is inhibiting using in vitro validations, that is ex vivo and in vivo analysis we have done for the confirmations. So the other area what we are working is on leptospira scan. So we are using AIML. We have using the images. So actually we are using the images for identifications of different leptospirosis. So whether it is a positive or negative leptospirosis, that can be done using this. Not only leptospirosis, you take any of the tool like any of the other pathogens. So any of the bacterial samples you can load here, and you can check how effectively that is identifying whether it is a gram positive, gram negative bacteria. If it is a gram negative bacteria, what are the possible treatment can be done? If it is a gram negative bacteria, what are the things has to be done? So where and all this particular analysis can be done is like here you can have towards the left you can see the pictorial representations in the public health survey. So smart cities, so rural and low resource settings, environmental monitoring. So there are different areas where we can use this particular leptospiro scan. And when it comes for the immunoinformatics, as I told you, like there are different publications you can do with this particular thing, immunoinformatics. One thing is like it's an easier component. Uh, the thing is like how you'll be planning, how what is the novelty you will be showing in that particular technique is very much important. You do any of the such kind of trace like either you can go for the whole genome sequences databases, or if you have a real-time data, then you can go for the NGS analysis, and then you can like identify the targeted protein using netopharmacological analysis. Then you can design the particular vaccine. It's depend on how novelly you will be presenting that particular component. The another important thing is I was talking about the adjuvants. When it comes for the adjuvants, you'll be talking majorly we'll be using the bioactive compounds, right? So this particular image represents the pictorial, like what is the traditional method. Till now we are using like we are taking the plant samples, we are going for different extractions, filtrations, purifying the compound, characterizing that particular compound, identifying what kind of compound it is. So this is a tedious process, right? So it's not like over a day or 1 or 2 days we'll be getting these results. If sometimes like even if it is not done on time, even this particular extract may loses its stability too. And most of the challenging is here is that how can all can select the novel medicinal plants. That is one particular challenge. Like everyone cannot take particular plant extract for their analysis, right? So, and when you come for the components, either it may be alkaloids, flavonoids, phenolic acid, terpenoids. Yeah, like those do have a multiple biological activity, like as Anisha told, repurposing drugs or screening of bioactive components for different targets. How they will be doing. So similarly, like there are different bioactive components which is already existed. Rather than going for a traditional isolation characterization process, you can select these kind of bioactive components. For that, one such tool, what our university is using is Pharmacodia Global and Cyber Drug Discovery. So here we have two different modules, that is Pharmacodia Global and Cyber Drug Discovery. So I will just showing you the tool, like how we'll be using those two tools for I hope the screen is visible.
Yes, Chandan, yes.
Yes, thank you. So when it comes for the Pharmacodia Global, here you can see the different modules. For example, what till now they have Bhargav and Anish talk about the screening of bioactive components for the targeted protein, or going for the repurposing drug. What and all the repurposed drug, which you can choose for your target of interest for inhibiting the particular diseases. So here, this particular tool, how it will be helpful for us is like, you can identify, this particular is a data source. So where all the data has been compiled in this particular software. Like if you have any target of your drug of interest, you can just enter your drug and you can go for the search. So we will be getting to know that where exactly this particular drug is working, and for which and all the target they are using this particular drug, whether this particular drug can also be used as an alternative for example, if you're working on a neurodegenerative diseases. Obviously, neurodegenerative diseases is like it has two components. One is anti-inflammatory and antioxidant component. Whether the drug of your interest has antioxidant and anti-inflammatory property, which someone is working somewhere else with the same kind of targets, so that can be done, that can be identifying using these particular tools. And also the targets, if you have your target of interest, you can just type here and you can go, when you go for the search, you'll be getting all these different tools like therapeutic areas, what and all the therapeutic areas you are concentrating, classification, either it may be small molecules, biological, vaccine, cell therapy, and like there are different approaches. So even if you want to take the approved drugs, the China, America, Europe, Japan, Hong Kong, so all the databases, even if you want to check the drugs which is right now in the phase clinical trials, starting from the phase 1 to phase 3 clinical trials, even that can be that data also can be get using this particular tool. So this is how the Pharmacodia Global will be helping us. So in order to support this Pharmacodia drug, we have another tool called as CyberAid drug discovery. So in the CyberAid drug discovery, we have two major components. One is CyberX lead. So let me open this CyberAid lead. So if you want to go for if you want to design your drug, or if you want to design your own novel compound, you can use this particular component called as cyber lead component. So here, let me take a project which we have already generated. So from the library, I'm taking the GLP-1. So here we are concentrating on the obesity. So how the GLP-1 receptors, how the target has been designed for GLP-1 receptors. So that is what we have taken. The components, the drugs, what and all you are seeing here is like, here you can see the clinical. So all these are approved drugs, and also you'll be getting some phase clinical drugs also. So what you can do is you can take these particular drugs. You can do the docking analysis. If that particular target is showing a better results, then you can validate with that particular results with your in vitro data, right? So for example, like if you want to go for novel drug discovery, what you can do is, whatever like chemistry peoples they are doing, like synthesis of a novel drug. So what they'll be doing, they'll be doing a scheme, right? So by changing the OH group, NH group, ortho region, and the para regions. So here similarly, what we can do is, by just alternating the ortho region or the para region, you can design your novel drug. So again, that can be validated using this particular CyberAid tool. Once your design drug is validated, then you can go for again the tool what and all we have talked, that is Schrodinger, or any of this, not only the Schrodinger, even like you can use there are different tools for validation, even that can be done. So once the validation is done, then you can check with the in vitro component, right? So this is all about the Pharmacodia CyberAid drug discovery. So yeah. Yes. So these two tools you can use for different component, as I told earlier. So if you have any doubts, you can let us know. No, this is Pharmacodia Global is not a free software. It's a paid software, what our university has purchased. Over to you, sir. Like, if anything.
And they have one more question from Sharma. Like, if we can take up the questions. Is there any AI tool for counting various types of blood vessels? So like they are using Image J, I think. Maybe probably for this, our team is using NGO tool, sir. Maybe
Angioton tool, what we are using. So it's a free tool.
It's a free
We
tool.
can use the angiotensin tool too.
For the angiogenesis study, we use the angio tool, wherein we can count the vessel lengths and junctions and density of the vessels can be calculated by using this angio tool. Maybe like this, we have so many other deep learning or maybe machine learning tools are available. Maybe you can check orbit image analysis are there, for the vessel segmentation and for the quantification, we have orbit image analysis. And maybe for the whole organ 3D vascular mapping can be visualized by using maybe a tool called as VESSAP, VESSAP. That can be used for like this. For the histology only, we have a visualizing tool called as cell profiler. You can use that also, sir. That is the answer for maybe
Yes. Thank you,
Shama
sir.
Shama ma'am.
Yes,
Maybe
thank you,
the
sir.
other question we had from Dr. Hemalata ma'am is which disease are currently the most promising targets for AI-based drugs? Particularly, ma'am, there is a lot of AI things are happening. Particularly, if I want to mention the disease areas, we are more concentrating on people are more concentrating, researchers are more concentrating on the oncology part. Again, cancer, that is large multi-omics data sets are being used nowadays. Maybe for the example, I can say KRAS, we can take our breast cancer, we can say, or maybe colorectal cancer. Maybe rare disease is one of the major disease area where AI component is used for the repurposing, I am telling. And for the neurodegenerative disease, already you have heard to Dr. Bhargav what complex disease biology which neurodegenerative disorders have got. For that, again, AD, PD, and MS, that is multiple sclerosis. These are the things which you can consider. Or maybe if any researchers looking for the metabolic disorders, like AI, artificial intelligence identifies the shared pathways across the diseases. Like say for this, I can take an example of type 2 diabetes, or maybe the obesity. In our team only, we are working on obesity. Again, for the autoimmune disorders or for the inflammatory diseases, also this network pharmacology, what Dr. Chandan D have discussed now, that also can be considered. That is the answer for your question, ma'am, Dr. Has Emalata, ma'am. Yeah, thanks for the appreciation which you all have given in the chat box. Thank you so much. And Saravanan sir is asking to share the PowerPoint presentation of session 1 and 2. Definitely, sir. Saravanan sir, definitely all the slides we will collect together, and we will be sharing it to the organizers. Maybe from them, you'll be receiving the information. And with this, I take this opportunity to thank the leadership and for the organizers for giving us this opportunity to present our whatever the thing which we know little to the participants. Thank you so much. Alone we can do so little, together we can do so much. With this, we will end this session. For any questions or any collaborations, please connect to us. My email ID is I'll be putting again in the chat box. You can connect to me or my team. My email ID is chandanes@jssuni.edu.in. Maybe other email IDs also I'll be sharing. Chandan D, that is my other speaker email ID. That also I am providing. Otherwise, Anisha Jain mail ID also I am forwarding. You can mail it to us if you have any queries or any collaborations. With this, I request Nivedita ma'am to end the sessions with the permission.
Thank you, sir. There's one more question, sir. For publication, there are prohibition and limitation to use the AI. So
For the
whether
publication, yeah, I missed it, I think. Just a moment, ma'am. Just a moment. I'll just
no
see.
problem, sir.
Where is it, ma'am? I'm just going through it. Yeah.
This is the question from Shama, ma'am.
Okay, for publication, there are prohibitions and limitation to use of AI. Yeah, definitely for the publication, the tools which we have discussed today, definitely this can be used to, say, for example, Pharmacodia, other cyber, those tools, definitely it can be used because they are the licensed versions of the tools that definitely can be used. Schrodinger or maybe whatever the AI tools which we discussed, definitely that can be used for to write the publication. If I understood perfectly the questions of Shama ma'am, that is for the publication, if you are writing the Scopus AI, if you are asking about Scopus AI, definitely that component which is generated through Scopus AI, the components can be taken and that should be rephrased, ma'am. That cannot be directly cut and we cannot do the cut and paste thing. Definitely the information can be taken from Scopus AI. What it is, does is maybe if time permits, I would have shown, but to Scopus AI, what it does is maybe based on the keywords, it gives us the proper information by taking the latest information. Latest information means from 2010 onwards, whatever the major outbreaks have taken from in that particular research area, that information, the major papers it will read. Maybe it takes some 10 to 20 seconds to read the articles, and maybe in 10 to 20 seconds only it generates the detailed report. That report can be used to write the papers, ma'am. Directly we cannot take and put it into our publications. Yeah.
Okay. Thank you, sir.
Thank you, ma'am. Thank you.
Okay, thank you, sir. As we come to the end of today's enriching session, I would like to express our sincere gratitude to Dr. Chandan and his team for sharing the valuable knowledge and expertise on artificial intelligence-driven bioinformatics approaches for modern drug discovery, from target identification to drug repurposing. Your insightful presentation has provided us with a deeper understanding of how artificial intelligence is transforming the landscapes of more modern drug discovery and has inspired us to explore new possibilities in research. I also extend my heartfelt thanks to our respected faculty members, research scholars, students, and all the participants for your enthusiastic presence and active participation, which made today's session truly engaging and successful. We look forward to your continued participation in the upcoming session of this faculty development program on artificial intelligence in transforming drug discovery research. Thank you all once again for joining us today. Have a wonderful day. Stay safe, stay curious, and happy learning. Thank you all.
Thank you. Thank you so much.
Thank you, sir.